Hello everybody, welcome to your science buddy. Today we're going to be taking a look at the cell structure. So these are the learning outcomes for today's lesson: cell biology, animal and plant cell common features, the differences between animal and plant cells, other types of cells, and at the end we shall look at end of chapter questions. So what is cell biology? Cell biology is the study of the basic structure and functional units of all organisms. So over here we have types of cells. We have two types of cells. We have prokaryotic and eukaryotic cells. Prokaryotic cells are simple cells and are much smaller than eukaryotic cells. They have no nucleus, rather they have DNA that floats, cellular DNA. They have no membrane-bound organelles. An example of this is bacteria. Next, we have eukaryotic cells. These cells are complex and are usually larger. They have a nucleus where DNA is stored and have membrane-bound organelles such as the mitochondria. Examples are plant, animal, and fungi cells. So over here we're going to take a look at the common features in the structures of plant and animal cells. If you take a look at the diagram, I have highlighted the most common and questionable features in red, the red line below. First we have the cell surface membrane, the nucleus, the chromatin, the DNA, the nucleolus, the cytoplasm, the organelles, which is everything within the cytoplasm, the mitochondria, the Golgi apparatus or complex, or as some people like to call it, the Golgi body, and the endoplasmic reticulum or the ER. So next we're going to take a look at the different cells, the differences between the features of the animal and the plant cells. So before we get into it, we can already see that the shape is different. We can see that the plant cell has a more defined shape than the animal cell, right? Next we have the nucleus. In the nucleus we have the nucleolus and the chromatin. Here are some diagrams displayed on the side. The nucleolus is the dark patch in the middle, and the chromatin in the second diagram, it's more visible, the thread-like structures. So the nucleus is the control center of the cell. It contains DNA and is surrounded by a nucleus envelope. That means a double membrane and has nucleus pores on the sides. The nucleus also controls cell activities like growth, metabolism, and reproduction. Next, we shall talk about the nucleolus. This is found inside the nucleus. Like I said, it's the deeply stained patch within the nucleus that you can see here in the diagram. It is found inside the nucleus and makes ribosomes, which help in production of proteins. It appears as a dark, dense spot, as I have described. Next, we have the chromatin. The chromatin are the thread-like structures that you see in the diagram over there. The chromatin are made of DNA plus proteins. They are found inside the nucleus. Uncoiled from DNA, condenses into chromosomes during cell division. Cell division is a chapter that is far ahead. Next, we have the cell wall. The cell wall is a strong, rigid outer layer in plants and in some bacteria and fungi. Note, the animal cells do not contain cell walls. Cell walls are mainly found in plant cells only. The cell wall is made of cellulose, which is tough and gives the cell support and gives it that shape that we talked about at the beginning. Yes, the well-defined shape. The cell wall also protects the cell from damage and stops the cell from bursting when it takes in water. This is during the process of osmosis. And here at the end we can see that the cell wall is not found in animals. Next we have the plasmodesmata. The plasmodesmata are sort of like pores that connect different cells. These tiny pores, we can say, allow two or more different cells to communicate with each other. So we can see that they allow substances such as nutrients and water plus signals to move between plant cells. It connects the cytoplasm of one cell to the cytoplasm of the other cell, helping them work together. Like I said, it's almost like a connection between two different cells. Next, we have the plant vacuoles. The plant vacuoles are larger than the animal vacuoles actually, and plants always have a large permanent vacuole, whereas for animals it is sometimes not present, which is why we call it a temporary vacuole, and it's usually smaller than the plant vacuole. In plants, vacuole stores water, nutrients, and waste. It also helps to maintain target pressure and can take up to 90% of the cell's volume, surrounded by a cell membrane called a tonoplast. This is basically like how we talk about a plant cell having a cell wall. Now the vacuole of the plant cell has a wall that we call a tonoplast. Next we have the animal vacuoles. These are many and sometimes absent, like I said, temporary vacuoles used for storage, digestion, and water removal. They are really not that questionable. They are not asked a lot during exams. I would advise you to take on plant vacuoles. Next we have the chloroplasts. In the diagram here we can see that the chloroplast is green. It's a green pigment. Basically it gives the plant leaves their green color. It is found in plants and not in animals. We are not green. We do not need the chloroplast because the chloroplast helps in photosynthesis and makes food using sunlight. The chloroplasts also have a double membrane and their own DNA. That means that they are able to replicate on their own. Next we have the endoplasmic reticulum. An easier way to remember is to abbreviate it as ER. This is acceptable during exams. The ER is divided into two categories. We have the rough ER and the smooth ER. The rough ER is covered with ribosomes and helps make proteins. It also sends these proteins to the Golgi apparatus, which we shall look at. Next we have the smooth ER. The smooth ER, as the name suggests, has no ribosomes and appears smooth in appearance. It makes lipids or fats and steroids plus hormones. It helps detoxify harmful substances. Next, we are going to take a look at the ribosomes. The ribosomes are the tiny dots that you see on the ER. So the ribosomes are the tiny structures that make proteins. They are found in all cells because all cells require proteins, plant and animal cells alike. Ribosomes are made of RNA and proteins. Where are they found? Ribosomes can be found attached on the rough ER, where they make proteins for export and use in the membrane, or can be found floating free in the cytoplasm. The function of the ribosomes, as we said, essential for making proteins, essential for growth and repair. Next, we're going to look at the Golgi apparatus or Golgi body or Golgi complex. It has several different names. The Golgi apparatus is found in both plant and animal cells. It is made of stacked, flattened cells like pancakes. It is often located next to the ER. You will notice that when ribosomes leave the ER in vesicles, these vesicles are taken to the Golgi body. These vesicles also help make up the Golgi body. The main function of the Golgi body is to modify, sort, and package proteins and lipids. It prepares materials for transport inside or outside the cell, produces vesicles or small sacs to carry the substance. You can see here in the diagram, we have the vesicles. These vesicles are the ones that transport the substances. You can think of the Golgi body as the post office of the cell. It gets proteins and delivers them. Next we have lysosomes. Lysosomes are commonly found in animal cells and rarely in plant cells. These lysosomes are small spherical sacs filled with enzymes. These enzymes help to destroy certain things. For example, there is damaged DNA in the cell. If the DNA of the cell is damaged, that means that the cell cannot conduct its functions properly. This means that the lysosomes will come in and will cause the cell to self-destruct. This will prevent the cell from duplicating or multiplying itself with its damaged DNA. So as we can see here, the functions to break down waste, old cell parts, and harmful substances, help with digestion inside the cell, also known as the cell's recycle center or cleanup crew. Next, we're going to take a look at the mitochondria, or mitochondria, it's my accent. The mitochondria are found in both plant and animal cells and are referred to as the powerhouse of the cell. You will notice that parts of your body, such as the kidney, the organs such as the kidney, have a large number of mitochondria because they require a lot of energy. So cells that require a lot of energy to perform their tasks usually contain a large amount of mitochondria. So we can see here that the mitochondria is the powerhouse of the cell and has a double membrane. The main functions: perform cellular respiration, converts glucose plus oxygen to ATP. Further ahead into the topics, you will look at ATP as a form of energy within cells. It has its own DNA, therefore it means that the mitochondria can reproduce on their own. Next, we have the cell surface membrane. The cell surface membrane or plasma membrane is found in all cells. Do not confuse cell membrane with cell wall. Cell membrane is found in plant and animal cells alike. Cell wall is found in plant cells and not in animal cells. So it is made of a phospholipid bilayer with proteins. We shall dive deeper into this phospholipid bilayer when we take a look at the cell membrane, which is a topic ahead. It is flexible and semi-permeable. The main functions: controls what enters and leaves the cell. That's what semi-permeable basically means. It controls certain things that enter, certain things that do not enter. Unlike the cell wall is fully permeable. That means anything can pass through. However, once it reaches the cell membrane, some things can go through, some things cannot go through. Provides some protection and structure. Involved in transport, cell recognition, and cell signaling. Next, we are going to look at the other cells besides plant cells and the animal cells. We have the bacteria and the virus. Bacteria are living single-celled organisms, the ones that we refer to above as prokaryotes, the ones that do not have membrane-bound organelles. So some extra features that they may have are the flagella, that's like a tail. We shall look at it in a diagram ahead. A tail that helps it move around. As I said, it contains circular DNA and no nucleus. That means that the DNA is freely floating within the cytoplasm. Can reproduce on their own through binary fission, and some can help while others cause diseases. Next, we have the viruses. Viruses are not living things because they need a host cell to reproduce. They cannot reproduce on their own, and they are much smaller than the bacteria cells. This means that they are also much smaller than the plant and animal cells, as we say that the prokaryotes are much smaller than the eukaryotic cells. Viruses are made of a protein coat plasmid and genetic material, DNA or RNA. No cell parts such as the cytoplasm or membrane, that means no cell-bound organelles. Reproduce by infecting a host cell, which is why we call it non-living, because it cannot reproduce on its own. Viruses often cause diseases. So here are diagrams, one of a bacteria cell, one of a virus cell. We can see on the bacteria cell that I have shown a part where some parts are absent while others are always present. We can see the parts that are always present in the bacteria cell are the cell wall, cell surface membrane, the cytoplasm, the circular DNA, and the ribosomes. By the way, these parts that I'm mentioning basically have the same roles and functions as the animal and plant cell parts. So if you wish to know the use of ribosomes, of the cytoplasm, or the cell wall, of the cell surface membrane, I advise you to take a look back up, pause the video, or rewind and check again. Then the additional features that are sometimes present, not always, as I say, the flagellum, the tail that helps it move, the capsule, which is usually present for additional protection. We have the infolding of the cell membrane, we have the plasmid, we have the pili. Then we have here the virus cell. As you can see, it's quite a very simple structure with a capsid, the protein molecules, and the genetic code. Now here are some exam style questions which you can take a look at. You can pause the video, answer them, and go through them and see if they are correct. What is the function of the nucleus in a cell? How is the structure of the cell different from the cell membrane? What is the main role of the vacuole in a plant cell? Describe the function of ribosomes. What is the difference between rough ER and smooth ER? What is the role of the Golgi apparatus in a cell? How do mitochondria provide energy to the cell? What is the function of lysosomes in animal cells? Explain what plasmodesmata are and why are they important? What does chromatin consist of and where is it found? What is a nucleolus and what does it do? List two structure features of prokaryotic cells. So these exam style questions will help you get ready for an exam or a test, and if you're still not confident enough, you can still go and research furthermore. Remember not to cheat yourself. Read through the questions, pause the video, and answer them. Thank you for listening.